Ivan Amato here. I'm the host of the Voices from DARPA podcast. Please note that in this episode, you might hear ambient noises such as birds, trucks, or even the hum of my refrigerator. That's because I have had to record my words in a home office as I play my social distancing role in our collective effort to minimize the spread of COVID-19.
Stealth technology, the internet, GPS in the palm of your hand, autonomous operation.
Technology is a driver of our times. Since its founding in 1958 in the midst of the Cold War, DARPA, the Defense Advanced Research Projects Agency, has been a driver of technology. Welcome to Voices from DARPA, a window onto DARPA's core of program managers. Their job? To redefine what is possible. My name is Ivan Amato, and I'm your DARPA host. I've been away since the end of 2018, and I'm excited to resurrect with this episode the Voices from DARPA podcast. However, I do this at one of the most trying times most of us can remember. Everything seems to have changed. Nothing feels right. On March 11th, one of the days on which I sat down with program managers in our recording studio, the World Health Organization, WHO, declared the infectious spread of the coronavirus as a pandemic. COVID-19, the disease caused by a new coronavirus that scientists refer to as severe acute respiratory syndrome coronavirus 2 or SARS-CoV-2. It has been among us at least since late last year when the first cases showed up in China. And now you and I and our 7.8 billion human brethren are living in pandemic times. The world's people are in this together. What we do know is that pandemics come and pandemics go. Depending on their severity and the collective response by us, they mete out differing amounts of human and economic loss. As an institution, DARPA has for many years recognized the enormous threats posed by emerging infectious agents. It has done so in its very DARPA way. Says DARPA as an institution, we are going to do something about this. In 2016, Brad Ringeisen, a civil servant working for the Department of Defense for nearly 20 years, became the deputy director of DARPA's Biological Technologies Office, BTO. In June, he upgraded to become BTO's director. Now he finds himself heading a team of program managers orchestrating a portfolio of technology programs that stand a chance of meeting this new coronavirus on its own molecular biological terms. These program managers are on their way to developing a technology platform that they hope will end the threat of pandemic disease, period. If they succeed, COVID-19 could become the last pandemic. This is not some Panglossian vision. This is hard science and technology that could take pandemic disease off of humanity's already long list of worries. BTO Director Brad Ringeisen.
DARPA plays a really important role for the entire government, quite honestly, but specifically for the Department of Defense, in its ability to predict what is emerging as threats to both our warfighters and our DoD personnel, but also to the country as a whole. And that has been something that's been persistent through the entire lifetime of DARPA. But specific to infectious disease and emergent disease, this is something over the last 2 decades, DARPA has been there. We've learned what is needed and what is required to be able to fight the types of outbreaks and pandemic that we're seeing right now.
Right now, it's the new coronavirus that is waking the world up to how devastating pandemic diseases can be. It was only a few years ago that we all experienced another wake-up call. That's when DARPA redoubled its efforts to investigate and develop technical means for quashing outbreaks of infectious disease before they can blow up into regional epidemics or global pandemics like the one that challenges us now.
Back in 2014, there was an Ebola outbreak. I'm sure we all remember that. That was the last time that we really started to worry about sort of an international pandemic. Because of sort of the limited transmissibility of that particular virus, requiring sort of bodily fluid transfer and things, that seemed to not be as contagious as the current coronavirus that we're seeing right now. But what that process did for DARPA, with some amazing program managers, amazing forethought that's happened back 5, 10, even 15 years ago, program managers started thinking about an ideal world where we could have a rapid response platform where we could, instead of having to develop vaccines over the course of years, maybe even decades in time, wouldn't it be great if we had a world where we could reduce that research and development process down to a matter of potentially weeks or months, rather than having to wait on that tremendously long time frame, which we know, living through what we're living through right now, it's just too long.
It's a combination of the nature of a new contagion, the speed at which a disease outbreak is detected, and public health and medical countermeasures that determines just how much and how fast and how far an initial outbreak will grow. Renee Wegersen, a program manager in the Biological Technologies Office since 2016, now runs several programs that she hopes will produce powerful new anti-pandemic tools, at least in time for the next threat. One of those programs, the DIGIT program, aims to improve our ability to detect any biological threat, known or new to humankind, anytime, anywhere. Her PREPARE program aims to leverage natural pathogen-fighting genes and proteins already in our bodies. Here's how Renee explains the difference between outbreaks, epidemics, and pandemics that drive her own work as a program manager.
The way you think about an outbreak is really just a anomaly in the number of diseases in a given region. Maybe for influenza, it's an increase in cases or a lack of seasonality, but something that is just inherently different from baseline would kind of constitute a outbreak. Beyond an outbreak, an epidemic is where you start to see more person-to-person transmission and a larger geographic region, even multiple countries being impacted. For something to be declared a pandemic, there's 3 strict criteria that have to be satisfied. So one is that in most cases this disease is novel, meaning that it's either something we haven't seen before or it's something that hasn't been around for a very long time. 1918 flu is a great example where there's very unlikely that there's immunity in the population. So effectively, many individuals, if not everyone, is at risk of this disease. That's the first criteria. And the second is there's person-to-person transmission. So it's not just simply animals to people in a zoonotic event, but genuine person-to-person transmission. And then finally, inherent in pandemic, it's as global. It's a pan-regional outbreak, which is now a pandemic. And those are the criteria that the WHO haven't set to declare a pandemic. And in fact, today, COVID-19 was declared a pandemic.
That was on March 11th, and in just a few weeks, the pandemic has terrorized ever more of the world. And it has amplified DARPA's sense of urgency to realize those innovations that will dramatically shorten the time from disease detection to delivering countermeasures at large scales. The agency runs a portfolio of programs that hit pandemics at every point possible, including even as dangerous viruses are mutating within animal and insect hosts into forms that can leap into people. If we could stop disease progression that early, says Brad Ringeisen, we would not see those upward shooting curves in the news depicting massive spread of infection and the devastation that comes with it.
In an ideal situation, you would really want to be able to move very, very much left of the curve that we are currently experiencing for the COVID-19 outbreak. We would want to be able to identify the emergence of diseases from animal populations. We'd want to be able to take those identifications early and detect maybe even in the animal populations, potentially even give countermeasures to those animal populations to be able to prevent the hop into humans that we've just witnessed over the last several months. So we have a program called Preempt that is exactly trying to do that.
This is where program manager Rohit Chichali comes into the picture. He has been a disease fighter for his entire professional life. Among his credits are co-establishing the global disease detection operations at the U.S. Centers for Disease Control and Prevention and helping to tamp down the spread of AIDS in South Sudan. And now he is focusing all of his experience and knowledge on the challenge of ending pandemic disease.
I came into DARPA in September of 2019, and in November I inherited a program called Preempt, Preventing Emerging Pathogenic Threats. And it really, what some people at DARPA call esoteric, but exactly what I think where we need to be going. It is the type of program that says we look at animals and we look at insects, say how can we stop the disease there before it spills over to humans? And that idea there is truly a prevention. And public health is all about prevention. And unfortunately, we like to say when we're successful, people don't know what we're doing.
Rohit says one of the best ways of achieving these hidden and unsung successes in public health and with battling pandemics is to understand how dangerous viruses arise in animals.
There has been a focus over the last 20 years on the concept of One Health. One Health brings together human and animal medicine and says that basically we know that animals actually are about responsible for 60 to 70% of human infectious threats. So, we think about things like Zika, dengue, yellow fever, chikungunya, Ebola. These have all come from animals. SARS, all three of these last three SARS in the last 15, 20, the last coronavirus of the last 15 years have come from animals. Well, then the idea is that this One Health concept says, let's look at animals so that we can get ahead of spread to humans. Can we prevent the spillover of a Hendra virus or a new coronavirus to people? Can we prevent that? Keep it in the bats. Keep it from coming into the intermediate reservoirs like the pangolins, the civet cats, for example, or the fowl, or the ducks and the birds in China, for example.
Understanding how these spillovers occur takes boots on the ground all over the world, Rohit says, and the performers, the investigators contracted to do research under his Preempt program, are leaving their boot prints behind far and wide.
Montana State is studying bats in Bangladesh, Madagascar, Ghana, and Australia, and they're studying bat ecology. They're trying to understand how do bats live, what do they feed on, when do they get stressed, when do they produce more in their urine, when do they produce more virus, and when does that virus spill over. So studying horses in Hendra, Australia, that name of the town is actually Hendra. That's where it was named from, the virus. So looking at where the spillover events have happened and then where the spillover events have happened for humans.
Once a spillover happens, then detection and diagnostics become pivotal pieces of the public health response. Brad Ringeisen explains a few of the ways his program manager colleagues have been moving the dial on this capability. One rapidly developing technology he points to will quickly identify, in say the sputum or blood of patients, the presence of molecular signs called epigenetic marks in the cells of a patient responding to a viral exposure. This would be like tracking the earliest footprints left by the viruses in people's bodies. You would know, for example, if and when people with common infection symptoms had been visited either by influenza viruses or SARS-CoV-2.
In DARPA language, we've very much tried to have foresight over the past 2 or 3 years to try to start developing detection platforms that are very easily adaptable to new diseases. Use gene editing technology, that there's lots of work that can be done just in computers to be able to simulate and to be able to identify the exact genetic code that you would need that would be specific to the type of emergent virus that we've seen now with the SARS-CoV-2. So we were able to immediately in January, just with genetic code information that was coming out about this virus, some of our performers were able to immediately identify very specific code that would specifically identify this virus, turn that into an assay that could then now be tested at the University of Nebraska, where there's many of these convalescing patients undergoing treatments in Nebraska. And so that was all done within a 2-month period of time to rapidly respond to now do the work that's necessary to be able to get FDA approval of that new assay for detection. That's one. And the second one that I'd like to talk about is a host response diagnostic program that we developed about 2 years ago called ECHO.
ECHO, run by program manager Eric Van Giesen, is an acronym that stands for Epigenetic Characterization and Observation. It's all about identifying the earliest signs in a person's body that he or she has been exposed to gravely threatening agents, including chemical, radiological, and biological weapons, or to natural threats like infectious pathogens. More than that, the handheld technology that Eric has been shepherding through development might be able to identify people who show no symptoms of infection, but nonetheless are contagious or likely to become so. It turns out that even early viral exposures leave telltale molecular marks on the DNA and proteins inside the nuclei of our cells. These are called epigenetic marks. They're a bit like graffiti tags on walls throughout a city. The goal of ECHO is to develop handheld devices that would be able to read those viral tags from easily gotten cell samples. Says Eric, the technology should be able to tell us if someone has been exposed to a particular virus, exactly when, and how that person is likely to respond to an exposure. Again, BTO Director Brad Ringeisen.
Where we're looking at the epigenetic response of your own body And that epigenetic response may occur on day one of exposure to that virus. And so your body starts adjusting, it starts adapting to that infection, and it starts leaving genetic marks on your genome. So we're very confident that we're going to be able to identify host epigenetic marks that are specific to this new COVID-19 outbreak. This is a program that we started 2 years ago. And again, it's starting to pay dividends now to be able to get very, very early, maybe even pre-symptomatic detection of a disease like this. And again, we're working with the University of Nebraska to be able to validate and to be able to test that assay as it develops and emerges and comes online.
Rohit Chitale's and Eric Van Giesen's fellow program manager, Amy Jenkins, has clocked several years working for DARPA, but only last June did she become a program manager in BTO. She didn't expect the ride would have accelerated quite this much so quickly and that the stakes would have climbed so high, but she is where she wants to be, furthering the power that the public health community can wield against disease.
And you always think you're ready until the real world scenario kind of drops itself in your lap. And then you realize, wow, there are a lot of things that we have to think about. There's a lot of coordination that has to go on. There's a lot of pressure. When you put out a program that says we're going to stop pandemics in 60 days and you have a declared pandemic on hand, people are looking at us to potentially make an impact on this outbreak.
Amy is overseeing a bevy of programs, including an especially comprehensive one called Pandemic Prevention Platform Program. Among the deliverables of her many programs are antibody-based treatments to stop infections early and long enough to buy time for the development of long-term protective measures such as vaccines. The goal of another one of her programs, called NOW, which is short for Nucleic Acids on Demand Worldwide, are mobile manufacturing platforms for producing new therapeutics against infectious diseases anywhere, anytime, and at scales that matter.
So my portfolio largely consists of how are we going to respond very quickly to infectious diseases. And the reason I think we need to respond quickly to infectious diseases is because we have seen it. We saw it in 2009 with H1N1. We saw it with Ebola, even though that was not respiratory and spread across the world. It's very easy to make the next connection to say if we had something that were spread by a respiratory disease, how quickly it could spread across the globe.
Central to Amy's portfolio is the quest to dramatically shrink the amount of time it takes to identify the antibodies in people who recovered from infections. Antibodies. These are proteins that our own immune systems invent when challenged by infectious agents and that are tailor-made to neutralize those agents. Amy and the researchers supported by her Pandemic Prevention Platform program are developing antibody treatments that could prove pivotal in slowing outbreaks before they can become epidemics and pandemics. The technology has made it beyond the concept stage, all the way to clinical trials, at least for a virus known as chikungunya. Amy says we could have protective antibodies against the new coronavirus in hand this year. This is how she chronicles the progress so far.
I am very interested in antibodies, and antibody technology provides a specificity. So antibodies are very, very specific for the pathogen that you are trying to combat, trying to target. They're made naturally by your body. So when you are exposed to, say, chickenpox when you were a little kid, your body made antibodies. Those antibodies then become memory B cells, and they hang out in your body. And if you ever see chickenpox again, they come back to life, if you will, and they stop you from getting infected by chickenpox. Well, we are very interested in that same type of technology. So when a person gets sick, they actually make antibodies. And so the thought with our program is that you can go in to a person that has recovered, and presumably their body has made antibodies that helped them recover from that infectious disease or that pathogen, and we can very rapidly find those antibodies. So once you find that antibody, it then becomes a matter of how do you give that thing that protected that one patient, if you're able to find the very good ones, and give it back to the rest of the population to protect them and prevent them from getting sick. And so that is really how we think about the use of antibodies. In many cases, we think of them as a fire break. So you give it to people so that they don't get sick. So that's the same kind of general concept that we want to use here. The problem that we have when we think about the use of these types of things is that we don't have decades to find that antibody, and we certainly don't have years or more to manufacture that antibody. And so that's where some of our other technologies come in. We have, as I mentioned, learned how to very quickly find the antibody. So we have certainly shrunk that timeline for finding the antibody. We also needed to invest in ways to ensure that we could manufacture that antibody much, much faster. And so traditionally, how they're made is they're made in giant stainless steel bioreactors inside mammalian cells. Traditionally, back in the 2014 Ebola outbreak, that process could take anywhere from 10 to 24 months to figure out how to get those cells to grow in that bioreactor and how to get them to make that protein properly. That was much too long for rapid response. And so we began investing in DNA or RNA technology. Antibodies are simply a protein. If we have the DNA or the RNA code for that DNA or RNA, if you inject it into your body, should make that protein, that antibody. We demonstrated that this was possible for a variety of different pathogens. So we looked at Ebola, we looked at influenza, we looked at multi-drug-resistant bacterial pathogens, we looked at chikungunya, Zika virus, so many different viruses, and generally always were able to demonstrate that, yes, this is a possibility in animals. We then turned our sights to humans. Through rounds of rigorous safety testing, we felt that it was time to potentially test this in humans. And we did do a human clinical safety trial. These types of technologies have been tested for safety in a phase 1 human clinical safety trial. And it has generally demonstrated the ability to inject, for example, an RNA intravenously into a human patient. That patient then, their cells take up that RNA, and their cells then produce the antibody that you asked, the code that you gave it to produce. And we actually completed that clinical trial with a company called Moderna as part of the ADEPT program in the fall of 2019. That was actually for a chikungunya virus, antibody against chikungunya virus. So we've demonstrated that you can use this genetic code, you can inject that into the person, and the person can become the bioreactor. So the thought is for this coronavirus outbreak, we may be able to actually manufacture antibodies in anywhere from 4 to 8 months.
As Amy sees it, this kind of antibody-based medical technology can change the pandemic equation in humanity's favor.
If you think about the fact that we could use antibodies, whether they be traditional protein antibodies that we injected, whether we inject the RNA or the DNA code, all of these could be used. You give them to healthy people. Those people are now protected for anywhere from a month to maybe as many as 6 months from getting disease. So if you think about this, we used to always talk about it in the context of the Ebola outbreak. I'll talk about it in the context of coronavirus now because the same thing potentially applies here. You have those people that have to respond. They are on the front lines. These are your nurses, your doctors, your first responders, the people that can't quarantine and stay home because they have to be in hospitals helping patients. These are the people that are potentially most at risk, so we call them our frontline healthcare workers, and this would be a population that it would make most sense to give this to initially so that they don't get sick, so that you keep your healthcare system functioning in a robust manner. Then you can also think about giving this type of, if you had enough of it, you could give this type of modality to people that have been exposed. So for example, if you have a family member, potentially all of their contacts could get this. So this is something that I think even 2 months ago, people maybe didn't understand, and people are now becoming much more comfortable with understanding contact tracing and what that actually means and why that's important. If you become sick and you tell your public health department, these are all the people I came into contact with, those are potentially now the people that are also going to get sick. You could then go to those people and potentially give them this so that they don't become sick. And now you stop these types of diseases from spreading. And it is frankly exactly how we have stopped some of the worst Ebola outbreaks in the history of Ebola outbreaks in the last 4 years. And it's going to be critical in this coronavirus spread as well, is just understanding who talked to who interacted with who. And we could certainly envision a situation where you use this type of technology to ensure that those contacts do not get sick.
Amy and her DARPA colleagues were able to swerve the research groups already working as performers in the Pandemic Prevention Platform Program, also known as the P3 program, to apply what they already have learned to the new COVID-19 pandemic.
So all of our groups are currently working on this, and we actually have groups that are currently, right as we speak at this moment, discovering antibodies against this coronavirus. We're able to obtain samples from a patient that, under informed consent, donated their blood to try to find some of these antibodies that helped that person recover. So several of our groups are currently working to try to identify those antibodies. I will caveat this by saying that this was one person and one person's response. And so we would typically do this with many people, maybe up to 10 people, because we just don't know how one person responds versus another. You want to have the best shots on goal. We had one person. Our groups are working extensively, literally around the clock, to try to identify those antibodies from that patient that may be the most useful for treating a broader swath of the population. It is going well. I am very cautiously optimistic. We have found initial hits that would indicate that we have some antibodies that do bind this novel coronavirus, but there is extensive testing that will go on in the next 2 to 3 weeks to really characterize and understand those antibodies. What we actually want to be able to do is neutralize the virus, or what we call neutralize the virus, which is prevent the virus from infecting human cells. So the first thing we'll do is we will test that in vitro, in a petri dish, if you will. Not exactly a petri dish, but in that type of setting. We will actually test and make sure that our antibodies block that pathogen from infecting human cells. Then the next step would be to then test it in animals, small animals or large animals, or both potentially. So we're talking mice or potentially all the way up to non-human primates. And if it looks like it can protect those animals from getting sick from this coronavirus, the next step would begin to manufacture it and get ready to test it for safety in a phase 1 human clinical trial.
This means all of this technology stands a chance of helping out even in this current pandemic, but it also sets the stage for disarming pandemic threats that humanity is sure to face in the future. Like some of her colleagues, Renee Wegerson is running programs that, if successful, could help solve the hard problem of quickly detecting any dangerous pathogen that might emerge. One of her ambitious programs is DIGIT, short for Detect It with Gene Editing Technologies. Imagine, she says, handheld devices that can detect any pathogen of concern with just about any biological sample, spit, blood, or urine, for example. That's what Renee wants to make so.
5 years from now, when we have all of our DIGIT devices up and running, first on the front end, we're going to have unparalleled biosurveillance capability where we can look at every patient sample that comes in and look across 1,000 different possible pathogens. In fact, envisioning every human pathogen that exists, if we just routinely look at that testing, that would be our opportunity to detect something very early, potentially even before an outbreak occurs. Once we have the biosurveillance hit, then we would want to get a diagnostic in place very quickly so that we could, okay, now we know there is something that's an anomaly, now rule in or rule out. Does this person have this disease or not? That's very important because you can help make a decision to quarantine somebody, but then also to give them the right medical countermeasure. So you want to make sure that you're matching treatment with the ailment, of course. That's sort of at the individual patient level, but then at the decision maker level, whether it's for a nation or a deployed unit, there needs to be decision making of, how do they respond and move forward? Is it rolling out medical countermeasures? Is it bringing your whole command back in? Whatever that might be, you actually could have, what we would call actionable information moving forward. So that would have been great. If we had this for this current outbreak, potentially we could have contained it before we hit this pandemic state. And that's really where we want to be moving forward.
Brad is all in on moving forward as fast as possible with the program managers in BTO and the innovation community they are nurturing. He feels their efforts really, really could reduce the consequences of future pandemic threats.
So if we step back and think about the next threat that inevitably is going to happen, I think what you see with the DARPA BTO suite of programs and suite of technologies that we're developing is that we really do see a pathway now where we won't have to go through the process of what we're going through right now to see this exponential growth, to see the danger of the threat to our population.
Brad points out that superlatively bold goals, like developing practical and deployable technology platforms that will intercept any infectious disease outbreak before it can threaten military readiness and large populations, takes solid partnerships throughout the innovation ecosystem. And it takes an institutional infrastructure that can deliver public health actions and medical countermeasures on massive scales.
We put a goal out to the community, we offer funding, research funding, and those researchers then go off and do the amazing work. But we can't take any of that work from the laboratory to actually used to be able to fight something like this without partnerships. And that's something that in my 3 years here at DARPA that I've really worked hard at, is trying to work with our DoD advanced developers, work with our HHS, NIH, ASPR, BARDA partners, because they're the ones that are really tasked with the advanced development and ultimately getting these types of products through the FDA and being able to actually distribute and manufacture them to get them out to the population. And so without those partnerships, and we're working with them literally almost every day during this outbreak to be able to make sure that these new innovations, these rapid response platforms, don't just die in the laboratory, that they actually make their way into the DoD personnel and to be able to protect the U.S. population as well.
So in this moment of shared anxiety and fear, it is a gift to imagine credibly of a not-so-distant future where no one has to worry that an outbreak of a new disease anywhere will spread into a devastating pandemic everywhere. It is heartening to know that there is a team of DARPA program managers who, with partners around the world, are making sure our most innovative researchers and technology developers can take those steps that will get us there. Thanks, listeners, for sharing this time with us. I hope you join us again for the next Voices from DARPA. Thanks also to Tom Shortridge and Ben Sullivan for help in producing this podcast. For more information about DARPA and the many breakthrough technologies the agency is working on, visit darpa.mil. And for links that enable you to download this and other episodes of the podcast, go to the Voices from DARPA page on DARPA's website.